Welcome to the course of Biomedical Electronics of final year Electronics and Telecommunication Engineering. Here we are going to discuss unit number 1, that is Introduction to Biomedical Systems. The very first unit in which you will be getting introduced to the biomedical instrumentation system and the acquisition of different biosignals generated in the different body systems. So let's see what are the contents of this unit. The first thing you will learn is the biomedical instrumentation system, the cell structure, biocell potentials, how these biocell potentials they are generated, the concept of bioelectrodes. These bioelectrodes are used to sense the biosignals or the biopotentials which are generated by different functioning of organs present in the human body. Then different types of bioelectrodes to measure biosignals, the transducers and sensors to measure biosignals like EEG, ECG, EMG, respiration, body temperature, the saturated oxygen level SpO2 and the pulse rate. And the very last part of this unit consists of artifacts that occur in biosignal acquisition. They include noise, power line interference, baseline interference, the concept of skin impedance, how to measure skin impedance, and the motion artifacts, and the techniques to reduce these artifacts that occur while we are sensing the biosignals from human body. The books that you can refer for this unit are Biomedical Instrumentation and Measurements by author Cromwell, which is of PHI publication, and the other book you can refer is Handbook of Biomedical Instrumentation by author R.S. Khanpur, which is of Tata McGraw-Hill. These two books you can refer for this particular unit number 1, Introduction to Biomedical System. Let's start discussing on this unit. So before learning this particular subject, you must be aware what is biomedical engineering and what is biomedical instrumentation. So basically, biomedical engineering, it is application of engineering principles and design concepts of medicine and biology all together. So whatever till now in the 4 years course of your electronics and telecommunication engineering, whatever you have learned, that engineering principles you need to apply to the concepts of medicines and biology, that all together forms biomedical engineering. And what is biomedical instrumentation then? So biomedical instrumentation, it is field of creating instrumentation that helps to measure, record and transmit data to and from the human body. As you all know, human body, it is a very complex engineering marvel which consists of various different types of systems inside it. Let's discuss about the different systems present inside the human body. And before learning the biomedical instrumentation and designing of different instrumentation systems, you must be aware about the human body and the different systems that are present inside it. And before designing any instrumentation system, you must know the anatomy and physiology of human body. But what is anatomy and physiology of human body? Anatomy, it represents the science of structure of human body. How is the structure of human body? That is described by anatomy. And physiology is nothing but science of functioning of body. Let's take an example of human heart. It has 4 walls. So what I am describing the anatomy of heart. And if I explain its functioning, like how the blood enters from inferior and superior vena cava inside the human heart, and then how it is circulated to whole body parts, the pure oxygenated blood, it is circulated to all the body parts through aorta. So this flow of impure blood entering into the heart, then going into the lungs for purification, and again taking that purified blood into the human heart and through aorta, spreading that blood to whole body part. What I am describing the function of that particular organ, that is heart, that represents nothing but the physiology. Human body, as I said, it is a very complex engineering marvel. It consists of various systems, as you can see in the figure. We have a circulatory system, that is cardiovascular system, the respiratory system responsible for inhalation and exhalation of air. There is nervous system, digestive system, reproductive system, muscular system, skeletal system, and many more other systems. See, in the engineering terms, this cardiovascular system can be thought of an hydraulic system. The respiratory system present in the human body, which is responsible for inhalation and exhalation of air, it can be thought of pneumatic system, the pressure of air coming in and going out. So it can be thought of pneumatic system. So we can relate all the human systems, human body systems, to the engineering concepts, and that all together forms biomedical engineering. And learning the anatomy and physiology, and then going for the development of the instrumentation system for the benefit of human, that is nothing but the biomedical instrumentation. So what is the role of biomedical instrumentation system? The basic objective of biomedical instrumentation system is gathering of information from different systems present inside the human body, doing the diagnosis, doing the evaluation, then after evaluating those signals, monitoring of that signal and controlling different parameters. That is the main objective of biomedical instrumentation system. Let's see different biomedical equipments. We will first get acquainted with different biomedical equipments present around. We have recording, measuring and monitoring equipments. In the hospitals, if you have seen, we have recording, measuring and monitoring systems. We have intensive care equipments, surgical and support equipments, imaging equipments like X-rays, CT scan, MRI. Then we have biotelemetry and communication systems, like if you want to transmit the data from the bedside monitor of a person in the hospital to some different location, we use biotelemetry and communication system. Testing and calibration equipments, these are very important in order to calibrate the existing recording, measuring and monitoring instrument present in different sections of hospitals. We have physiotherapy equipments, electrotherapy, radiotherapy equipments. These are very broad categories of different biomedical equipments which are available. Let's see the instruments related to blood. We have blood cell counter, which you can find in the clinical laboratories of hospitals. We have blood pressure meter, we have blood pH meter. You can see the figure of blood pH meter. It is used for measuring the pH value of blood. Blood flow meter, digital BP meter, that is blood pressure meter. This you might have seen all around in your homes also. You have the digital blood pressure meter, the flame photometer, spectrophotometer. These are various instruments related to the measurements done on the blood or the flow of blood inside the body or outside the body, like taking the samples outside and doing the measurements. Then we have electrocardiograph, which is related to the heart, and these are the different heart instruments which I am showing you right now. Electrocardiograph, that is ECG, pacemaker, defibrillators you might have seen in hospitals, which are useful for giving the temporary shocks to the patient in order to regulate their action of heart. These defibrillators and pacemakers, they are the same thing. If your heart is not working properly, to regulate its function, we use this two category of instrument, that is pacemaker and defibrillator. Then there is plethysmograph, phonocardiograph. Phonocardiograph, it is related to listening the sounds of different functioning of heart, the opening and closing of the heart, and the opening and closing of heart creates sounds. If we want to diagnose any problem related to the opening and closing of valves, if they are closing and opening, is there any delay in that, so to check that we go for the phonocardiograph. It is audio test of your heart. Then electronic stethoscope, you know stethoscope, you can see the figure. Now let's see the brain instruments. Brain instruments include your EEG, electroencephalograph, the tomography, that is CT scan. You can see in the figure, the first figure shows you the ECG monitor, and the second one is the tomography, the CT scan setup. Related to muscles, we have muscles instruments. The first category includes the electromyograph, which is the recording of activity of the muscles. It is called as EMG. You can see in the figure the EMG is carried out, and you can see it over here. The second is muscle tomograph. Instruments related to kidney, we have dialysis machine, lithotripsy. Related to eyes, we have oculometer, you can see the figure, and the aid for blind. Let's see different monitoring instruments. We have biomonitor, bedside monitor. In ICUs, you might have seen there is a monitor, and different electrodes are placed on the human body, and continuously the signals are given to that system which is present besides the bed of human where that patient is lying. So that is nothing but a bedside monitor. If you have visited hospitals and the ICUs, you might have seen the systems exactly besides the patient's body, the monitors which are showing you the signals. There are nothing but the bedside monitors. We have fetal monitor. Fetal means the small infants. So we have fetal monitors. An imaging instrument is also an important category of biomedical instruments. We have radiography, that is X-rays. In the figure you can see the first figure is showing you the X-ray. Second is magnetic resonance imaging, MRI, the setup of MRI you can see here. Then we have CT scan and fluoroscopy. These all are the imaging instruments. Ultrasound, you can see the machine of ultrasound over here, and echocardiography, and nuclear medicine such as PET. These all are the imaging instruments. Okay, now I think the different biomedical instruments which are present in any of the hospital that I have shown you, and now we will see the biomedical instrumentation systems block diagram, or it is also called as man instrument system. Before proceeding to biomedical instrumentation system, I would like to tell you that there are two different kinds of measurements done. One is vivo and the other one is called as vitro. So what is difference between vivo and vitro, I will be telling you. Vivo means the measurements that are made on or within the human body are nothing but vivo measurements. For example, like a device inserted into the blood in order to measure the pH of blood, that is nothing but your vivo kind of measurement. And vitro, the opposite of vivo is vitro. Measurement is performed outside of the human body. That is, if you take the sample of blood and then make the measurement of blood pH, then that is vitro kind of measurement. So what is vivo and vitro? Vivo is the measurement done within the human body, and vitro is the measurement performed outside the human body. Now let's see the different blocks in the biomedical instrumentation system. The subject, you can see subject is nothing but your human body on which the measurement is to be done, and in some of the cases the stimulus, external stimulus are required. The stimulus may be like visual, a flash of light, it can be auditory, that is the tones can be allowed to pass through the human body, they can be tactile, or direct electrical stimulation of some part of nervous system. Then we have different transducers placed on the subject. This is device which is capable of converting one form of energy or signal to another. These transducers may measure temperature, pressure, flow, or any other variables that can be found in the body, but its output is always an electrical signal. Two or more transducers, they can be used simultaneously to obtain the relative variations between the phenomena occurring in the body. The next come your signal conditioning equipment. The part of instrumentation system that amplifies, modifies, or in other way changes the electrical output of transducer is the signal conditioning equipment. Signal conditioning equipment, it is used to combine or relate the output of two or more transducers. The input and output of the signal conditioning equipments, they are electrical signal, although greatly modified with respect to input. The main purpose of signal conditioning equipment is to process the signal from transducer in order to satisfy the function of the system and to prepare signals suitable for operating the different displays or the recording equipments that it follows. So we have display equipment. The electrical output of this signal conditioning equipment must be converted into a form that can be perceived by one of the man's senses and that can convey information obtained by measurement in meaningful way. The input to the display device, it is modified electrical signal from the conditioning equipment. Then we have recording, data processing and transmission equipment. It is often required to record the measured information for the latter or to transmit it from one location to another. Where automatic storage or processing of data is required or where computer control is employed, an online analog or digital computer may be present. Recordings can be of two types: using graphic pen recorder, which is used to produce a paper record of analog waveforms, or storing on the magnetic tape recorder. We have one more block, that is control. So whenever it is necessary or desirable to have automatic control of stimulus, transducer or any part of the man instrumentation system, a control system, it is incorporated. It usually consists of a feedback loop in which some part of output from the signal conditioning or the display equipments, it is taken and it is used to control the system in some of the way. So this is the block diagram of man instrumentation system or the biomedical instrumentation system. And from a very long time I am seeing biosignal. So what is basically biosignal, that we will see the definition of biosignal. A biosignal is any signal in the living being that can be continuously measured and continuously monitored. Biosignals can be of two types: active biosignals. Active biosignals are nothing but body generated signals. They can be electrical or non-electrical biosignals. The electrical biosignals can be EEG, ECG, EMG, EGG, ERG, and non-electrical biosignals, they can be temperature or blood pressure. And the other type of biosignal is passive biosignal. It is body modulated signal, for example X-ray. So to have an X-ray of the skeletal part of human body, we need to expose the X-rays on the body, and then that is the imaging technique by which we produce the X-ray. So it is passive biosignal is body modulated signal. An external stimulus, that is X-ray, is applied to the skeletal part of the body to get the image of the skeletal structure. Coming to different sources of biomedical signals, we will see the sources in detail. The biosignals are those signals, as I told you, they are used primarily for extracting the information on biological system which is under investigation. And biosignals, they convey us very useful information related to the phenomena that is happening in the body. The process of extracting information could be as simple as feeling the pulse of a person on the wrist, or as complex as analyzing the structure of internal soft tissues by an ultrasound scanner. Bioelectric signals, these are unique to biomedical systems. The first source of biomedical signal is bioelectrical signal. They are generated by the nerve cells and the muscle cells. The basic source in the cell membrane potential, which under certain condition may be excited to generate an action potential. We will see the whole process of bioelectrical signal generation, like how action potential and the resting potentials they are generated by the cell. The electric field generated by the action of many cells constitutes the bioelectric signal. The most common example of bioelectric signal are the EEG, that is electrocardiograph, and EEG signal, that is electroencephalograph signal. The other source of biomedical signal is bioacoustic signal. The measurement of acoustic signal created by any biomedical phenomena provides information about the underlying phenomenon. The example of such signal are flow of blood in the heart, as I earlier told you, through the heart walls, and the flow of air through the upper and lower airways in the lungs, which generate typical acoustic signal. So the best example of measurement of bioacoustic signal is your phonocardiograph. I told you earlier when we were discussing the introduction of this particular unit. The next is biomechanical signal. The signal originate from some mechanical function of the biological system. They include all types of motions and the displacement signal, pressure and flow signal, etc. The movement of the chest wall in accordance with the respiratory activity is an example of this type of signal. We have other category of source of biomedical signal, that is biochemical signal. The signals which are obtained as a result of chemical measurement from the living tissues or from the samples analyzed in the laboratory. The example, the pH value of blood is the example of biochemical signal. Many other examples we can see. The example are measurement of partial blood pressure of carbon dioxide, that is SpCO2, partial pressure of oxygen, that is SpO2, and the concentration of various ions in the blood. These are the examples of biochemical signals. Now coming to what I told you is bioelectric signal, that is the main focus of our whole course of biomedical electronics. So for that, first we need to learn the cell structure and then see the bioelectric potentials, how they are generated, the action potential and the resting potential. The whole discussion will be on that part. Let's start with the cell structure first. So you can see in the figure, the human cell structure is shown. The cell, from Latin cella, meaning the small room. So the cell is the basic structural, functional and biological unit of all the known organisms. A cell, it is the smallest unit of life, and cells are often called as the building block of life. There are many different types, sizes and shape of cells in the body. Basically the cell consists of three parts. The cell membrane, you can see here in the purple color, the nucleus, and between the two is the cytoplasm. You can see here. So cell membrane we will discuss first. Every cell in the body is enclosed by a cell membrane. The purple color you can see is the cell membrane. The cell membrane separates the material outside the cell extracellular from the material inside the cell, that is intracellular material. It maintains the integrity of cell and control the passage of materials into and out of the cell. The materials within the cell must have access to the cell membrane, the cell's boundary, for the needed exchange. Nucleus we will discuss. Nucleus, it is formed by a nuclear membrane. You can see around a fluid. Nucleoplasm is the control center of the cell. Nucleus is the control center of the cell. Nucleus contains DNA, the genetic material of the cell. The nucleus determines how this cell is going to perform, and as well as the basic structure of cell, it is decided by the nucleus itself. Then the last is the cytoplasm. The cytoplasm is, you can see here, this skin colored cytoplasm. Cytoplasm, it is a gel like fluid inside the cell, and it is the medium for all the chemical reactions that are taking place. It provides a platform upon which other organelles can operate within the cell. All the functions of cell expansion, cell growth and the replication, they are carried out in the cytoplasm of a cell. Yes. Now coming to the origin of the bioelectric potential. So we know the structure of cell. Now we will see how the bioelectric signals they are generated. So the system in the human body, it generates its own monitoring signal when they are carrying out different functions. The signals which are generated, they provide us useful information about the function they are carrying out. Bioelectric potentials that are usually the ionic voltages produced as a result of the electrochemical activity of certain cells. For that purpose, we learned the cell structure, and everybody knows transducers we saw in the biomedical instrumentation system. So these transducers, they are used to convert this ionic voltages into the electrical signals for the further signal processing and the displaying of the signal that is being generated by the different systems present in the human body. Coming to the bioelectric potential generation, again, a cell, it consists of an ionic conductor separated from outside environment by a semi-permeable membrane. I showed you the membrane, cell membrane, which act as a selective ionic filter to some of the ions. That means some of the ions that can pass through the membrane freely, whereas others cannot do. So this is the property of the semi-permeable membrane, that it allows only certain kind of ions to pass inside the cell, while it blocks certain kind of ions outside the cell. All living matter is composed of cells of different types. You know, human cell may vary from 1 micron to 100 micron in diameter, from 1 millimeter to 1 meter in length, and they can have typical membrane thickness of 0.01 micron, so very small. Surrounding the cell of the body are the body fluids, which are ionic and which provides a conductive medium for the electric potential. So the principal ions, they are sodium, potassium and chloride, Na+, K+, and Cl-. The membrane of excitable cell, it permits entry of potassium and chloride ion, while it blocks the entry of sodium ions. As I told you, the cell, it is surrounded with a semi-permeable membrane. Then that semi-permeable membrane allows only certain kind of ions to pass, while it blocks certain ions outside the cell itself. So the membrane of excitable cell, it permits entry of potassium and chloride ion, potassium, that is K+ and Cl- ions, but it blocks the entry of sodium ions, that is Na+ ions. So Na+ ions will be outside the cell, and the potassium K plus and Cl minus chloride ions, they will be allowed to pass inside the cell. Now you can imagine the outside of the cell we have positive ions, and inside the cell we have the chloride ions which are negatively charged. So inside of the cell we can see it is negative than the outside of the cell. So the membrane potential is called as resting potential, and it is always measured from inside of the cell with respect to the body fluids outside the cell. So outside of the cell, as we saw, it is having the sodium ions which are positively charged, and inside of the cell it has chloride ions which are negatively charged. And if we measure the potential inside with respect to the outside body fluids, you can see it will be negative. So the resting potential of cell is negative. You can see the interpretation. Inside of the cell is negative, outside we have positive sodium ions. So if we measure from inside with respect to outside of the cell, this potential will be negative, and that is nothing but your resting potential. And this process is called as polarization of cell. The cell in the resting state is called as polarized cell. The resting potential ranges from minus 60 millivolts to minus 100 millivolts. So this is the typical range of resting potential. Now if this cell is excited, the membrane, it changes its characteristics. The sodium ions, now they start rushing into the cell. The characteristics of the membrane, it changes, and the sodium positively charged sodium ions, they try to come inside the cell, and the potassium ions, you can see in the figure, they try to go outside. So same is written here, the sodium ions are rushed in the cell, and at the same time the K plus potassium ions, they try to move from inside to outside. Sodium ions rush into the cell, at the same time the potassium ion try to move from inside but unable to move as rapidly as the sodium ions. So we have the chloride ions negatively charged outside, and the concentration of positively charged potassium and the sodium ion concentration will be more. So you can see the inside of the cell will be more positive than the outside of the cell, and if we measure the potential from inside with respect to outside, the potential will be positive, and that potential is nothing but action potential, and the typical value of action potential is 20 millivolts. And this process is called as depolarization of cell. Now cell comes from the depolarized state to again polarized state when the equilibrium is achieved, and that is nothing but called as repolarization of cell. You can see again the same membrane potential is achieved. This cell again comes to the repolarized state. Here you can see the electrical activity associated with one contraction in a muscle is shown. From the resting potential, the stimulus is applied, it comes into the excited state, polarized to depolarized, and again getting repolarized. So the cycle you can see here, it is shown for one contraction in a muscle. Now we will see recording of electrical activities associated with different system in the human body. So various electrical bioelectric signals are generated by different systems in the human body. So the recording of electrical activity associated with the functioning of heart, it is known as electrocardiogram. I was saying ECG, EEG, EMG, ERG, EGG. So what is that? So ECG, it is recording of electrical activity associated with functioning of heart. Then we have electroencephalogram. It is nothing but the measurement of electrical activity or recording of electrical activity of your brain. The human brain, it generates rhythmical potentials which originate in the individual neuron of the brain, and this potential gets summated as millions of cell discharge synchronously and appear as a surface waveform. And this recording of electrical activity of the signal generated by the neurons is known as electroencephalogram. We have EMG. The contraction of skeletal muscles results in generation of action potential in the individual muscle fibers, and a record of which is known as electromyogram. Simply stated, it is the recording of electrical activity associated with the movement of muscles. Then we have ERG. ERG is nothing but electroretinography related to your eyes. The biological source for the measurement of ERG signal is eyes. For EGG, EGG represents electrogastrogram related to your digestive system. So stomach is the biological source for the measurement of bioelectric potential generated by your digestive system. So these are different electrical signals and the recording of electrical activity associated with the different systems present in the human body. So for ECG, we have biological source as heart. For EEG, brain. For electromyogram, your muscular system. For ERG, we have eyes, and for EGG, digestive system is the biological source. So we are discussing bioelectric signals. So basically the ECG, it has a frequency range of 0.05 to 120 megahertz and the signal amplitude of 0.1 to 5 microvolt, and it is measured by using the skin electrode. Electroencephalography, EEG signal, it has frequency range of 0.1 to 100 hertz. Signal amplitude is from 2 to 200 microvolt, and the signal can be sensed by using the scalp electrodes. You know, electrodes, they are capable of converting this body's ionic potentials into the traditional electric voltages. Electromyogram, EMG, it has frequency range of 5 to 2000 hertz and the signal amplitude range is from 0.1 to 5 microvolt, and basically to get an accurate result we go for the needle electrodes. Electroretinography, as I told you earlier, ERG, the frequency range is DC to 220 hertz, signal amplitude of 0.5 to 1 microvolt, and the electrodes that can be used are the contact electrodes. We have EOG, that is electrooculography. The frequency range is from DC to 100 Hz, signal amplitude of 10 to 3500 microvolt, and the electrode used are contact electrode. So we saw what these bioelectric signals are and how the bioelectric signals they are generated, the action potential, resting potential of cell, how the cell goes from a polarized state to depolarized state, then again from depolarized state to repolarized state, that is nothing but going to the normal state of the cell. All this phenomenon we have seen, and this bioelectric signals they are generated by the different systems operating inside the human body, like your human's heart, the brain, the neurons present in the human brain, then the muscular contraction and relaxation, that also too results into the bioelectric signals. But how to sense this bioelectric signals? These bioelectric signals they are sensed with the help of bioelectrodes. So the bioelectric events that have to be picked up from the human body before they can be put into the amplifiers for the subsequent recording or displaying purpose, the signals they need to be captured from the human body. So device that is converting the ionic potentials into electrical potentials are called as electrodes. You might have learned this in your chemistry. This is the basic definition of electrodes. But what are bioelectrodes? So transducers that are converting the body's ionic current into electronic current flowing in the electrode is nothing but a bioelectrode. Bioelectrode, it functions as an interface between the human's biological structure and the electronic system, where the processing of the signal takes place and then the signal is recorded or it is displayed on a monitor. Electrodes, they play an important part in the satisfactory recording of bioelectric signal, and their choice requires a careful consideration. They should be comfortable for the patient to wear for a long period, and they should not produce any artifacts. That is, the bioelectrodes, they must not produce any kind of noise in the signal recording. So another desirable factor is the convenience of the application of electrode. That should also be considered while choosing the electrodes for any measurement of bioelectric signal. Bioelectrodes should have this properties, that is, they should be good conductors, they must have low impedance. If the impedance will be low, so the CMRR of the amplifier, it can be maintained higher, that is, the noise or the common signal coming to the amplifier, it can be reduced. They should not polarize when the current flows through them. The bioelectrode should establish a good contact with the body, and they do not cause any motion. If motion is caused, then the motion artifacts, they get introduced. Metal should not be toxic. If it is toxic, it will cause harm to the human. They should not cause itching, swelling or discomfort to the patient. Like if the measurement is to be for a longer duration of span, if patient is suffering from a severe disease, he needs to be under continuous monitoring. So the electrodes will be placed on the body for a very longer period of time. So they should not cause itching, swelling or discomfort to the patient, and they should be easy to clean. If they are reusable, they should be easy to clean, and they should also be mechanically rigid. So there are various electrodes used for measurement of electrocardiogram, that is your ECG signal, for EEG signal, electroencephalogram signal, then for the electromyogram signal, ERG signal, EGG signal, EOG signal, and many other signals. All other non-electrical biosignals can also be measured with the help of transducers and the sensors that you can learn in this unit number 1. So we will stop here. Thank you. I hope you understood what is discussed in this video. Happy e-learning.